Hello Pasangala, Vanakkam and welcome. This is your master teacher Nivetha making bio learning very simplified just for all of you guys. So இன்னைக்கு நம்ம செஷன்ல பார்க்க போறது part 3 of principles of inheritance and variation, that is nothing but your sex determination, mutation and pedigree analysis. இந்த மூணு டாபிக் தான் இன்னைக்கு நம்ம செஷன்ல பார்க்க போறோம். So before I get into the session, I just wanted to give you a clarity. நேத்து organism and population நான் அப்லோட் பண்ணியிருந்தேன். அதுல ஆமா adaptations உங்க new NCERT syllabusல இல்ல. Okay, that means that is not included in your CBSE 2024 exam. Okay, I apologize for not actually checking it. Okay, வாங்க இன்னைக்கு செஷனுக்குள்ள போலாம். But populations இருக்கு, understanding ecosystem is very important. So அது வந்து ஒரு basic idea கொடுக்கும் உங்களுக்கு அந்த சாப்டரை இன்னும் பெட்டரா புரிஞ்சுக்கிறதுக்கு. Okay, yes. So we have Vati's top questions. We have very simple 5 questions that will be asked by the end of the session. இன்னைக்கு செஷன்ல இருந்து 5 சிம்பிளான questions curate பண்ணிருப்பேன். Check it. Do answer it and test yourself immediately. So vaanga session kulla pogalam. So as I already discussed, sex determination, mutation and pedigree analysis will be today's agenda. Okay, so what is sex determination? Okay, an organism, whether it's a male or it's a female, what is going to determine the sex of an organism is called as sex determination. So மூணு விதமான sex determination இருக்கு. ஒன்னு வந்து you have special chromosome called sex chromosome which is going to determine the sex. Okay, and you have haplodiploidy. Haploidy na N, diploidy na 2N. If an organism is haploid, it will develop into male. If an organism is a diploid, it will develop into female. It's very interesting. And in certain organisms, it's though very rare, okay, the environmental factors determine the sex of an organism. Okay, so most of it, okay, 80% of the time, the sex is determined by sex chromosomes. Okay, and who discovered this X chromosome? It was Henking in the year 1891. Okay, very important. இப்போ நம்ம chromosomal method of sex determination பத்தி பார்க்கலாம். So மூணு இருக்கு, XXXY type, XXX0 type and ZZZW type. Okay, so very important 5-mark question. Sex determination, explain the different types of sex determination using chromosomal method. Okay, so modala let's talk about this XXXY type. So XXXY type na enapa? So the people who have XY will develop into male, right? And the people who have this X chromosome will develop into female. Okay, so who is the heterogametic sex here? The male is the heterogametic sex pasangla. Okay, so the male produces 2 different kinds of gametes. 50% of his gametes will have X chromosome and the other 50% of his gametes will have Y chromosome. Very simple concept yet very important. Okay, next XXX0 na enna? The male is heterogametic. Okay pasangla, and there is only one X chromosome. ஒரு X chromosome இருந்தா அவங்க male, 2 X chromosome இருந்தா அவங்க female. Okay, then you have ZW type. இங்க என்னன்னா ZW is your female. So here in this case the female is heterogametic, the male is homogametic. Okay, இந்த மூணுமே ரொம்ப clear-ஆ படிக்கலாம். Okay, as I told you, male heterogamety. So where do you see male heterogamety? XXXY type. Okay, and you also see it in certain insects. Okay, XXXY type of sex determination ரொம்ப ஈஸியா ஞாபகம் வச்சுக்கணும்னா human beings ஞாபகம் வச்சுக்கோங்க. Okay, so here the determining sex is your male. Okay, the male is heterogametic. அதுதான் இங்க கொடுத்திருக்காங்க, heterogametic, and the female is homogametic pasangla. Okay, now coming to female heterogamety. So adula enna solrom? ZZZW type. Okay, here the female produces 2 types of gametes. Okay, whereas the male produces single type of gamete. So inga enna? Male vandu homogametic. The female is heterogametic. Okay, just opposite avladha. And you have one more special type of sex determination, that is your XO type of sex determination. So XO type na enna pa? 2 X chromosome irunduchna. Okay, if there are 2 X chromosome, it is going to develop as a female. Okay, if there is only one X chromosome, it is going to develop as a male. Simple, romba easy. Where do you see this XX, X0 type of sex determination? You see it in grasshopper. Romba important MCQ question in your exam. Where do you see XO type of sex determination? Okay, these are the three important types. This is your XX, XO type. Okay, so you can see 50% of them will be, there are chances. If there is a organism, there is 50% chance that the organism can be female, and there is another 50% chance that the organism can be male, seen in grasshopper. Okay, next, this is this sex determination in human beings. Okay, so here what, the mother is homogametic, the father is heterogametic, right? XX, XY, XX, XY. So what do you see here, pasangla? There is 50% chance that the child could be a male. There are 50% chance that the child could be a female. So who is determining the sex? This Y chromosome is determining the sex. Who is producing this Y chromosome? Male. So it is the males who are responsible for the gender of the child. Okay, whether it's a male or a female. Rombo simple. Okay, then you have your ZZ-ZW type, we already discussed. Okay, Inga, the female is heterogametic. Okay, the male is homogametic. So here who is deciding the sex? It is basically your female. You're supposed to draw the Punnett square and then write it very nicely. Okay, where is it seen? It is seen in birds, it is seen in butterflies and moths, and also in certain fishes. புரிஞ்சுதா பசங்களா? WZ டைப்னு சொல்லுவாங்க இல்ல ZW டைப்னு சொல்லுவாங்க. 5 mark question, sex determination. Okay, now we have the second method of sex determination, that is haploid diploidy. ரொம்ப simple. Haploid na enna? N number of chromosome. The haploid organism will develop into males. Okay, the diploid organism will develop into females. Okay, இங்க ரெண்டு விதமான females இருக்கு. ஒன்னு வந்து queen, another one is a worker. Okay, the queen is the leader of that swarm or a hive. நீங்க பார்த்திருப்பீங்க bee hive பார்த்திருப்பீங்க கரெக்ட்டா? அதுல ஒரே ஒரு bee நீங்க ரொம்ப கேர்ஃபுல்லா அப்சர்வ் பண்ணிருந்தீங்கன்னா you can see one bee that is very huge, and she is called the queen, and queen is fertile. She can reproduce. Okay, whereas my worker, though she is diploid, she is sterile. She cannot reproduce. That's the whole point here. Okay, so let's see how this haploid diploid, 3 mark question, explain haploid diploid sex determination in honeybees. Okay, it's very commonly seen in honeybees. Okay, so honeybees la or a diploid cell la 36 chromosomes irukum pasangla. Haploid na enna? It will have 16 chromosomes. Okay, so what will happen? The queen bee will undergo gametogenesis. She will produce eggs. Correct? So when she produces eggs, what will happen? They all will be diploid. See how beautiful it is. And haploid egg, okay, without fertilization, develops into a drone. Okay, develops into what? Male. And this process of development without fertilization is known as parthenogenesis. என்ன சொல்றாங்க பசங்களா? Parthenogenesis ன்னு சொல்றோம். Okay, development of male without fertilization, development of male in order, development of an organism without fertilization of the egg is called as parthenogenesis. Romba important question. Okay, now there is fusion. This is going to develop into a female bee. In female bee, there are 2 types, workers and queens. You guys might be wondering, how can a worker become a queen? So one of the worker who is very strong will be selected as a queen. Okay, because this queen feeds on a special substance called royal jelly. Okay, or queen sagamode, she will leave a little bit of royal jelly for the next queen. Okay, when the royal jelly is consumed by the queen or is consumed by the worker grows in size and becomes queen. See how interesting. Okay, you guys would have heard honeybees are the best socially organized and insects. they are beautifully organized. Okay, அவங்க hive பாத்தீங்கன்னா அவ்வளவு அழகா ஒரு 2-3 நாள் அவங்கள அப்சர்வ் பண்ணீங்கன்னா அவங்க எப்படி வேலை பார்க்கறாங்க, how they divide their work, everything will be so beautifully understood. Okay, so இத தான் நம்ம haploid diploid type of sex determination ன்னு சொல்றோம். Okay, so ரெண்டு மெத்தட், ஒன்னு வந்து chromosomal method, இன்னொன்னு வந்து haploid diploid method of sex determination. இப்போ நம்ம பார்க்க போறது mutation. Ma'am, mutation na enna ma'am? Mutation is a sudden, ரொம்ப important ஆன definition, sudden heritable change. என்ன change பசங்களா? Heritable change in the DNA. Okay, or in the sequence of the DNA, we can call it as mutation in the sequence of DNA. So என்ன நடக்குது பசங்களா? இது வந்து உங்க நார்மல் ஜீன், உங்க நார்மல் புரோட்டீன், இங்க ஏதோ ஒரு மியூட்டேஷன் நடந்துருக்கு, ரைட்? So that is going to change the entire protein sequence. So ஒரு abnormal protein synthesize ஆகுது. Okay, and this mutation was studied by a person called Hugo de Vries. He was the father of mutation, and he also had T.H. Morgan, Thomas Hunt Morgan. So along with your linkage, he also studied mutation. Okay, so this Hugo de Vries studied mutations in a special plant called Enothera lamarckiana. We call it as evening primrose. Okay, so he observed 834 mutation. எத்தனை mutation? 834 mutation பார்த்தாரு. Okay, in around 54,343. இந்த நம்பர்ஸ் எல்லாம் ஞாபகம் வச்சுக்கணும்னு இல்லை, just see the curiosity. Okay, எவ்வளோ பொறுமை இருந்திருக்கணும் இந்த மனுஷனுக்கு 54,000 plants படிக்கணும்னா. Yes or no? So that's very important to understand here. So the credit of discovery of mutation is given to your Morgan. So Morgan enna padicharu? Mutation padicharu. Adukapra linkage pathiyum padicharu. So he discussed and he described both pasangla. Okay, he observed some white-eyed male Drosophila in a population of red-eyed Drosophila. இந்த red eye ன்றது ஒரு wild type, அது எப்பவுமே இருக்கக்கூடிய ஒரு phenotype. Okay, சடனா அந்த red-eyed populationல ஒரு white-eyed population வந்துருச்சு. ஸோ அதை வந்து அவர் mutant phenotype ன்னு சொன்னாரு. என்ன சொன்னாரு பசங்களா? Mutant that doesn't originally belong to them. ஏதோ ஒரு புது கேரக்டர் வந்திருக்கு. How did that new character arise? Because of mutation. ஸோ அதனால தான் அவர் அந்த white-eyed மேல வந்து mutant ன்னு சொன்னாரு. Okay, so in Drosophila, eye color is sex-linked character. அப்படின்னா என்னப்பா? This eye color, the gene for the eye color is present on my X chromosome. Okay, அதுதான் நான் வந்து sex-linked character ன்னு சொல்றேன். Gene for red eye is dominant over the gene of white eye. So in the red eye வந்து dominant character, white eye வந்து recessive character. And I had one more person called Muller. Okay, so Muller discovered the induced mutation. என்ன ma'am? Induced mutation na enna ma'am? Me, a human being, can artificially induce mutation. நான் mutations கொண்டு வர முடியும். அதுதான் நான் induced mutation ன்னு சொல்றேன். And this induced mutation can be induced using help of radiations, X-rays, UV rays, gamma rays. All these radiations cause mutation. Okay, ரொம்ப முக்கியமான விஷயம். Okay, now let's try to understand what is a mutation. Mutation in புரிஞ்சது. Okay, so we have a mutator gene and a mutable gene. Okay, this gene called as your mutator. என்ன சொல்றாங்க இத? Mutator gene. So this mutator gene induces mutation on another gene. That gene is called as a mutable gene. Okay, so ஒரு gene வந்து இன்னொரு gene mutate பண்ணும். அந்த mutation induce பண்ற gene வந்து நான் mutator ன்னு சொல்றேன். And the gene which gets mutated is called mutable gene. Very simple. And I have a term called muton. What is a muton? It is a unit of mutation. Okay, we have replicon. It's a part of your DNA which undergoes replication. Similarly, I have muton. Okay, very important. It's a unit of mutation. இப்போ நம்ம முக்கியமா பார்க்க போறது மியூட்டேஷன் வந்து ரெண்டு விதமா இருக்கு பசங்களா. ஒன்னு வந்து chromosomal mutation, இன்னொன்னு வந்து gene mutation. Okay, so mutations are of two types. முதல்ல நம்ம chromosomal mutations பத்தி பார்க்கலாம். It is a change that occurs in the morphology of chromosome. So how these chromosomes are, அதுல ஏதோ ஒரு change கொண்டு வரது தான் உங்களோட chromosomal mutation. Okay, resulting in the change in the number or sequence of a gene without any change in the ploidy. The ploidy வந்து அப்படியே தான் இருக்கும். Diploid எல்லாம் change ஆகாது. If it is diploid, it will remain diploid. Okay, there can be one extra chromosome or one chromosome which is deleted. இல்ல அந்த chromosome குள்ளேயே there can be deletion, duplication, அந்த மாதிரி நடக்கலாம். Okay, so ரெண்டு விதமானது இருக்கு. Heteroploidy or genomic mutation. It is a change in the number or the set of chromosome. Okay, it is of two types, euploidy and aneuploidy. Okay, so heteroploidy நம்ம genomatic mutation ன்னு சொல்றோம். It is a change in the number of sets or chromosome in sets. Okay, அந்த குரோமோசோம்ல இருக்க சேஞ்ச் தான் நம்ம வந்து இப்படி சொல்றோம். ஸோ அது வந்து ரெண்டு விதமா இருக்கு, euploidy and aneuploidy. Okay, euploidy na it is an addition or deletion of one chromosome, ஒரே ஒரு chromosome. Okay, and aneuploidy na it is change in the entire set of chromosome. இப்போ 2n இருக்கிறது 4n ஆகலாம், 4n இருக்கிறது 8n ஆகலாம். That is what I call it as aneuploidy பசங்களா. I hope it is all clear, euploidy and aneuploidy. Okay, now let's move on. So chromosomal aberration, chromosomeல நடக்கக்கூடிய நாலு முக்கியமான types of mutation: deletion, inversion, duplication and translocation. So let's try to understand one by one very quickly. So what is deletion பசங்களா? Deletion is the loss of a part of a chromosome or the entire arm of the chromosome is deleted. அததான் நான் வந்து deletion ன்னு சொல்றேன். ஸோ இந்த deletion வந்து ரெண்டு விதமான deletion இருக்கு. ஒன்று வந்து terminal, இன்னொன்று வந்து intercalary. Ma'am, terminal na enna, intercalary na enna? This picture is itself going to explain you. நீங்க அந்த நோட்ஸே பார்க்க தேவையில்லை. Terminal deletion means the loss of the terminal of the chromosome. The loss of segments from the ends of the chromosome is called as terminal deletion பசங்களா. ends-ல இருந்து ஏதோ ஒன்று delete ஆகுது, அதை தான் நம்ம terminal deletion ன்னு சொல்றோம். இப்போ intercalary நடுவுல இங்க பாருங்க, இந்த 3-4 delete ஆயிருக்கு, கரெக்டா? இங்க 1-2 delete ஆயிருக்கு, இங்க 3-4 delete ஆயிருக்கு. That is what we call it as terminal deletion. So example for deletion, 𝐃𝐢𝐬𝐳 syndrome. Okay, so what is this 𝐃𝐢𝐬𝐳 syndrome? This is extra information da. Okay, so there the children who have this 𝐃𝐢𝐬𝐳 syndrome, there is a loss of a part of your chromosome. Okay, fifth chromosome la oru part வந்து it is lost. Okay, so what happens due to this? There will be abnormal larynx development. So anga alugumbodhu that cry will be like a cat like cry. Okay, so that is why we call it as 𝐃𝐢𝐬𝐳 syndrome. Okay, so deletion la rendu vidham, terminal and intercalary. Okay, அடுத்தது I have inversion. What is inversion பசங்களா? Listen to the definition very clearly. It is a breakage of chromosomal segment but reunion on the same chromosome in the reverse order. There are of two types, paracentric and pericentric. Okay, so என்ன நடக்குது பசங்களா? இந்த part of my chromosome is broken. Okay, first break பண்றாங்க, break பண்ணி அதை திருப்பி உல்டாவா வந்து அதை fix பண்றாங்க. So this 6-7 has become 7-6. That is what we call it as inversion. Okay, இப்போ 5, 6, 8, 9 ன்னு இருக்குன்னு வச்சுக்கோங்க. Okay, just some random numbers I have written. Okay, now what is happening? If this part is removed, okay, and then it is inserted in the opposite direction, that is what we call it as inversion பசங்களா. இந்த inversionல ரெண்டு விதமான inversion இருக்கு. ஒன்று வந்து paracentric inversion, இன்னொன்று வந்து pericentric inversion. Paracentric inversion na they involve your centromere involve ஆகாது. Only one arm is involved, one chromosomal arm is involved and there is no centromere which is involved. அததான் நான் வந்து paracentric ன்னு. அதே இங்க pericentric ன்னு பாருங்க, two arms are involved. Two arms are involved and there is an entire transposition. You can see how the movement is happening. Okay, அததான் நான் வந்து pericentric ன்னு சொல்றேன். Pericentric involves your centromere and it involves two arms. Paracentric does not involve your centromere and it involves only one single arm. Okay, very important for NEET exam. Then duplication, rombo easy. Okay, one part of your chromosome is duplicated. Okay, அது அப்படியே இன்னொரு காப்பியா டூப்ளிகேட் ஆகுது. And this can result in homozygous recessive conditions. Okay, so you have a bar I character that is observed in your Drosophila that is due to duplication in your X chromosome. So என்னென்ன படிச்சிருக்கோம்? முதல்ல நம்ம deletion படிச்சிருக்கோம், inversion படிச்சிருக்கோம், அப்புறம் duplication படிச்சிருக்கோம். இப்போ நாலாவது type of chromosomal aberration, that is your translocation படிக்க போறோம். So what is translocation? General Englishல translocation னா என்ன? ஒரு இடத்துல இருந்து இன்னொரு இடத்துக்கு shift ஆகுறது தான் translocate ஆகுறது. கரெக்ட்டா? So what is actually happening? The part of the chromosome moves from one chromosome to another chromosome. அதை தான் நான் வந்து translocation ன்னு சொல்றேன். This is not like your crossing over. Crossing over is exchange. Okay, and it happens between homologous chromosome, and this is happening between non-homologous chromosome. Remember, and this happens due to mutagen. So crossing over into the vital process. This is not a non-vital process. It can many a times be very, very harmful. It can even lead to cancer like lymphoma. Okay, so enna nadakudhu pasangala inga? What do you actually understand? You can see this part of your chromosome is shifting from here to here. Okay, and these two are non-homologous chromosome. Other than a simple translocation in solren, I have the next translocation, interstitial or shift translocation. So you can see they are shifting in interstitial. Interstitial na in between. Okay, inga pathina they are translocating at the ends. At ends. Okay, here you can see they are translocating in the between. Reciprocal na enappa? It is actually transporting inga irundhu inga, anga irundhu inga, mutual. I give you, you give me. That's it. Okay, it is the exchange of segment between non-homologous chromosome. So மூணு விதமான translocation இருக்கு. என்னென்ன பசங்களா? Interstitial translocation, reciprocal translocation and simple translocation. So this completes your chromosomal aberration, insertion, deletion, okay, inversion and translocation. Okay, now let's move on to point mutation. So in the point mutation நம்ம என்னன்னு சொல்றோம்னா, உங்க gene mutation இருக்குல்ல, gene mutation are of two types. One is point mutation, another one is frame shift mutation. Okay, we call it as two types of mutation. So point mutation na enna pasangala? These are due to structural changes in the DNA molecule. Ennoda DNA molecule changes agudhu. Why? It occurs at a single locus at one point. What is locus? A point where an allele is located or a gene is located is called as locus. These alter the message that is conveyed by a gene. Okay, this point mutation is also called as single nucleotide polymorphism. அது அப்படியும் நம்ம சொல்லலாம். Okay, ரொம்ப ரொம்ப முக்கியமான விஷயம். Yes, what happened? Yeah. Yes, so gene mutations are sudden and distinct changes in the gene which can be detected by visible changes in the phenotype of an organism. So these point mutation could be X-linked, so we call it as an X-linked disorder or a mutation, or it could be autosomal. Yes, so rendu vidha irukku, substitution and frame shift. Substitution na enna pasangala? One nucleotide or one base is substituted by another base. Okay, so you being substituted by another person, or teacher varati and the teacher ku substitute anupuvanga, adhe da idu. Okay, so substitution is of two types. One is transition, another one is transversion. Okay, enna solranga? Transition and transversion. So transition na enna pasangala? What do you understand by transition? Anybody? It is changing of a purine to purine, okay, or a pyrimidine to pyrimidine. ஒரு பியூரின் வந்து பியூரினா தான் மாறும், ஒரு pyrimidine வந்து pyrimidina தான் மாறும். So A to G, okay, or T to C and vice versa. Okay, this is what we call it as transition. Transversion na a purine to pyrimidine or a pyrimidine to purine. This is very, very important. Okay, it causes a change in the genetic code which leads to change in one amino acid in the structure of a protein. ரொம்ப முக்கியமான example, sickle cell anemia. Sickle cell anemia is a beautiful example for substitution. Okay, so let's try to understand what happens in sickle cell anemia. Okay, this is U. Okay, 𝐾𝐤 is becoming 𝐃𝐢𝐬𝐜. One base, one single nucleotide base changes the entire life of a person. Correct? So 𝐃𝐤 will code for glutamate. Okay, whereas 𝐃𝐢𝐬 codes for valine. And this happens in my beta globin gene. Okay, and as a result, the beta globin protein is faulty. And என்னோட biconcave இருக்க வேண்டிய RBC வந்து crescent shaped ஆயிருது, sickle shaped ஆயிருது. Okay, as a result, they become anemic. So sickle cell anemia பத்தி நம்ம இன்னும் டீடைலா டிஸ்கஸ் பண்ணலாம் in the next class. Okay, yes. Now, frame shift mutation, shift of the frame. Okay, like the mutations are very harmful as compared to your substitution. Okay, so which are the chemicals which basically induce frame shift mutation? Acridin, proflavin. Okay, they all cause this induced mutation. Okay, they are going to result in the structural changes of my DNA. So how is that going to happen? I'll give you one best example. You can see here. This is a normal translation. இதுல வந்து எந்த mutationுமே கிடையாது. Okay, இங்க என்ன நடக்குதுன்னா I'm adding one nucleotide. Okay, I'm just adding one nucleotide here. Somewhere in between I'm adding the entire change in the reading frame. Okay, is called as frame shift mutation. So எப்படி நான் வந்து இதை define பண்ணலாம்? Addition of a nucleotide, changing the entire reading frame. Okay, changing the entire reading frame is called as frame shift mutation. Okay, that is what we call it as frame shift mutation. So here you can see one nucleotide is deleted, here you can see one nucleotide is added, and you can see the entire change in amino acid sequence. ரொம்ப முக்கியமான விஷயம். Okay, and one such example for a frame shift mutation is cystic fibrosis. It's a very complicated disease pasangla. So in the cystic fibrosis la enna nadakuduna, there is a mutation in the CFTR gene. Okay, so CFTR endradhu enadhu? Cystic fibrosis transmembrane conductance regulator gene. Okay, so this gene is going to help in the transmission of substances in your plasma membrane. It's going to form a transport protein. But in the gene when the mutate ஆயிடுச்சுனா, it is not going to work properly. அப்ப என்ன ஆகுது பசங்களா? The entire alveoli is filled with mucus. Okay, and they will find it very difficult to even breathe. Okay, so that is your cystic fibrosis, one of the example for your frame shift mutation. இப்போ நம்ம mutagens பத்தி பார்க்கலாம். Mutagens na very important 3-mark question. Mutagens are those which causes mutations. The agents which bring about mutations are called as mutagens. ரெண்டு முக்கியமான mutagens இருக்கு. ஒன்னு வந்து physical mutagen, இன்னொன்னு வந்து chemical mutagen. Physical mutagen includes all your radiations. Okay, radiations வந்து ரெண்டு விதமா பிரிக்கப்பட்டிருக்கு. ஒன்னு வந்து ionizing radiation, இன்னொன்னு வந்து non-ionizing radiation. So what is this ionizing radiation? You have alpha, beta, gamma, X-ray. They are all ionizing. Okay, you have your UV rays which is non-ionizing. Okay, so these UV rays have become a potent, very potent mutagen, because nowadays we are all exposed to a lot of UV rays. Okay, why? Because there is an ozone layer depletion, and that's resulting in more and more UV rays to penetrate into the Earth's atmosphere. Okay, then you have chemical mutagens. They are more harmful than radiation because the body is not protected against chemicals. The source of chemical mutagens are the food we eat, the air we breathe, and the water we drink. Correct? And also the other cosmetics that we apply. Everything that you are exposed to are examples for chemical mutagen. Example: mustard gas, okay, carbon tetrachloride, and nitrous acid. These are some of the examples of chemical mutagens. Okay, so inga paakalam radiation, UV rays, X-rays. Okay, then I have chemical, cigarette smoking. Okay, 80% of the people who are chain smokers have more probability of getting cancer in their future. That is, it could be lung cancer, it could be buccal cavity cancer or oral cancer. எப்படிப்பட்ட cancer வேணா அவங்களுக்கு வரக்கூடிய probability அதிகமா இருக்கு. Okay, and you have nitrate and nitrate preservatives which is used in all your fancy fast foods. Okay, that can also be a mutagen. And benzoyl peroxide in the bleaching எல்லாம் பண்ணுவாங்க, facial bleaching, அதெல்லாம் peroxide என்ற ஒரு content is also a potential mutagen. Okay, other than that we also have infectious agents like human papilloma virus, okay, HPV virus, and this HPV virus causes cervical cancer. என்ன மாதிரி cancer cause பண்ணுது பசங்களா? cervical cancer cause பண்ணுது. And this cervical cancer க்கு இப்போ ரீசெண்டா சில மாதங்களுக்கு முன்னாடி they have discovered a vaccine for this cervical cancer which is given free in all the government hospitals. Okay, and even in private it's not so expensive, 350 to 400. And this cervical cancer affects the cervix of women. Okay, so all those women above the age of 30 or 35 can take this vaccine and prevent themselves from cervical cancer. And I also have Helicobacter pylori, okay, which actually spreads through contaminated food. It can also cause mutations. Okay, so these are the different kinds of mutagens. இப்போ நம்ம last part of the lesson வரோம், pedigree analysis. Ma'am, pedigree na enna ma'am? It's very simple. It is a graphical representation. எப்படி சொல்லலாம் பசங்களா? Graphical representation of a trait. Okay, representation of a trait is called as pedigree. Okay, so how a trait has been inherited. இப்போ person X க்கு ஒரு genetic condition இருக்குப்பா, இந்த genetic condition உங்களுக்கு யார்கிட்ட இருந்து வந்துச்சு? அம்மா கிட்ட இருந்து வந்துச்சா, அப்பா கிட்ட இருந்து வந்துச்சா, இல்ல ரெண்டு பேர்கிட்டயும் அந்த alleles இருந்துச்சா? சரி, அப்பா கிட்ட இருக்கு, இந்த அப்பாக்கு யார்கிட்ட இருந்து வந்துச்சு? Whether did he inherit from his mother or from his father? So it is going back and tracing how this inheritance pattern could have happened is called as pedigree analysis. Okay, நம்ம இந்த chapterல we are talking about disorders. Okay, disorders மற்றும் நம்ம வந்து you really can't find out. Okay, you can find out any trait, any character. இந்த ஏழாம் அறிவுன்னு சொல்லி ஒரு சூர்யா மூவி வந்திருக்கு, கொஞ்சம் பழைய படம் தான், அதில் பார்த்துருப்பீங்க. So we all carry the genes from our ancestors, alleles from our ancestors. Okay, it just needs to be induced, right? The same thing we can actually understand here. So it is an inheritance pattern of a particular trait represented in the family tree over generations is called as pedigree analysis. So நாலு விதமான pedigree இருக்கு. ஒன்னு வந்து autosomal recessive, இன்னொன்னு வந்து autosomal dominant, X-linked recessive and X-linked dominant. Okay, so in autosomal na என்ன பசங்களா? The trait which is carried by your autosome. நான் ட்ரேட்னு சொல்லும்போது அது ஒரு டிசீஸாக இருக்கலாம், இல்லை அது ஒரு கேரக்டராக இருக்கலாம், அதையும் கிளியராக ஞாபகம் வச்சுக்கோங்க. ட்ரேட் which is carried by autosomes is called as autosomal trait. Okay, X-linked na என்ன பசங்களா? The trait which is carried by X chromosomes, அதை தான் நம்ம வந்து X-linked சொல்கிறோம். டாமினன்ட்னா என்ன மேம்? ரெசசிவ்னா என்ன மேம்? டாமினன்ட்னா என்ன பசங்களா? ஒரே ஒரு allele போதும், only one allele is enough to cause the disease ன்னு சொல்லலாம், disorder ன்னு சொல்லலாம், or to exhibit the trait. Okay, say for example I have capital A small a, so this person is having only one capital allele and that allele is more than enough to cause the defect. ஓகே, ஆனால் ரிசெசிவ்னா அப்படி கிடையாது பசங்களா, ரெண்டு அலீலுமே இருக்கணும், இந்த மாதிரி ஸ்மால் ஏ இருக்கணும், ஸ்மால் ஆர் இருக்கணும். ஒன்லி in the recessive condition they will be able to exhibit the trait. Okay, and the same thing holds good for X-linked recessive and X-linked dominant. Only thing is the alleles are located on the X chromosomes. Okay, இப்போ நம்ம the most commonly used or difference between recessive and dominant பார்த்துடலாம். Okay, recessive na enna pasangla? The trait often skips generation. Ena only if both the parents are carriers, only then the child could be affected. Okay, so a trait usually does not skip generation. I'll show it to you with the help of a pedigree. Okay, affected individuals will always have affected children. If na affected na, all my children will be affected. Okay, then affected individuals may have normal children. Okay, so that's very important. இதுதான் உங்களோட ரொம்ப ரொம்ப முக்கியமான விஷயம், the symbols used in pedigree. Okay, if you draw a square, it is a male. If it's a circle, it is a female. Okay, if we draw something like this, that means the sex is unspecified. They are not sure whether it was a male or a female. Okay, அப்படி எழுதி அதை உள்ள வந்து நம்ம ஷேட் பண்ணோம்னா it means they are affected individuals. Okay, so when I draw a line like this, it means it is mating. Okay, இதே ரெண்டு லைன் draw பண்ணனா it means it is consanguineous marriages. So what is consanguineous marriages பசங்களா? Anybody? It is marriage between close relatives. Okay, so இப்படி draw பண்ணி இப்படி எழுதி, if I show the progeny, it means the progeny is there. Okay, இந்த progeny நான் shade பண்ணனா it means the progeny is affected. Okay, so இப்படி எழுதி அதுக்குள்ள number எழுதுனா it means there are 5 affected offsprings. Okay, so in your exam, in your NCERT, they have given 2 important pedigrees. One is an autosomal dominant trait, the pedigree A, and pedigree B is for an autosomal recessive trait. Okay, so autosomal dominant trait. Now I will explain you everything. So imagine this is generation 1. Okay, this is generation 2. Okay, and this is 2. This is generation 3. Has the generation skipped any of the generations? ஏதாச்சும் ஒரு generation ஆது has this skipped? No. So we call it as a dominant trait. So அதனால வந்து நான் dominant trait ன்னு சொல்றேன். Okay, now is the male and the female, are they both equally affected? ரெண்டு பேருமே ஈக்குவலா தான் அஃபெக்ட் ஆயிருக்காங்க. It's not like only the males are affected or only the females are affected. It's not like that. Both the sexes are equally affected. So we call it as an autosomal, என்ன சொல்றோம் பசங்களா? Autosomal dominant trait ன்னு சொல்றோம். Both the sexes are equally affected. So we call it as a autosomal dominant trait. Okay, இன்னொரு முக்கியமான விஷயம் என்னன்னா, example for autosomal dominant trait, though it is not mentioned in your book, டெக்ஸ்ட்ல கொடுக்கல, டயக்ராம்ல கொடுத்துருக்காங்கல்ல, மயோட்டோனிக் டிஸ்ட்ரோஃபி இல்ல மஸ்குலர் டிஸ்ட்ரோஃபின்னு கூட சொல்லுவோம். மேம் இந்த மஸ்குலர் டிஸ்ட்ரோஃபினா என்ன மேம்? நம்ம மசில்ஸ்ல ஒரு டிஸ்ட்ரோஃபின்னு சொல்லி ஒரு புரோட்டீன் இருக்கும் பசங்களா, and that dystrophin is what helps in contraction and relaxation of muscles. But in people with myotonic dystrophy or muscular dystrophy, the dystrophin protein or the dystrophin gene is mutated. So அந்த dystrophin protein வந்து ஒழுங்கா produce ஆகாது, அதனால அவங்களோட muscles வந்து ஒழுங்கா contract ஆகி relax ஆகாது. So என்ன ஆகும்? உங்க lungs, உங்க stomach எதுவுமே work ஆகாது, because they are all muscular in nature. Okay, and as a result they die at a very very young age, and that's your myotonic dystrophy பசங்களா. Okay, இப்போ இதை வந்து ஒரு குட்டி cross மாதிரி போடலாம். So imagine this person is normal. Okay, and this person could be homozygous or could be heterozygous. Okay, homozygous ஆவும் இருக்கலாம், heterozygous ஆவும் இருக்கலாம். நம்ம அந்த person heterozygous ன்னு எடுத்துக்கலாம். So when I take it as heterozygous பசங்களா, so what will be the conditions? AA, AA and AA. Yes or no? This can happen. So this person could be AA, this person could be AA, this person could also be AA, this person is AA. So the people who are carrying the dominant allele will be affected, the others will not be affected. Romba simple. Yes or no pasangla? Ippo this person is also normal, so I don't see anyone who is affected. Okay, now this person is this, the same thing, so you go and again affected. This people will be AA. So using alleles, I am just showing you what has actually happened. Okay, so this is your autosomal dominant trait. இப்போ இதே மாதிரி let's try to understand autosomal recessive trait. Okay, so autosomal recessive trait will take sickle cell anemia. Okay, will take SS, SS. Okay, ரெண்டு பேரும் carriers இருக்கணும். Though they are not affected, they carry this recessive allele. Okay, என்ன நடக்கும்? S small s, S small s, S capital S, S small s, S small s, S small s. What do you see here? You can see 25% of the progeny is actually affected. Amava illaya pasangla? So you can see this is SS, this is SS, this is capital S, okay, and this person, should be a carrier because இங்க ஒரு affected இருக்கு. Okay, this person should also be a carrier. இது ஒரு புது person, இவங்க வந்து இந்த progeny relate ஆகல. Okay, they are marrying, the person who is getting married. Okay, and this could be a normal S capital S. So என்ன நடக்குது? Again these both are carriers, so they will be affected. Okay, so autosomal recessive trait க்கு மூணு example இருக்கு, that is sickle cell anemia. Okay, then you have phenylketonuria and thalassemia that is involved in your syllabus. Okay, so இந்த மாதிரி probability based பண்ணின question ஒரு pedigree கொடுத்துட்டு they can ask you to identify what kind of pedigree it is, or they can ask you to identify what is the probability that person X or person Y could be a carrier, person X or person Y could be a normal person. அந்த மாதிரின questions நிறைய expect பண்ணலாம். Okay, soon, very soon I will solve numericals based on this chapter. ரொம்ப முக்கியமான numericals கண்டிப்பா இந்த chapterல இருந்து solve பண்ணலாம். So இந்த chapter ஓட last part ஒன்னு இருக்கு, that is genetic disorder and chromosomal disorders, which I will be doing in the next class. So do stay tuned for more bio learning, and don't forget to attend Bhathi's top question and test yourself immediately what you have learned. Thank you so much.